Hello students, welcome to EPG Pathshala. Myself Monica Seni from Department of Anthropology, University of Delhi. Today I am going to discuss on the module Cytogenetic, Immunogenetics and DNA methods to solve the paternity disputed cases under the Forensic Anthropology paper. Students will be able to define and describe the term cytogenetics. This module will be able to define and describe the DNA methods. Students will be able to know cases of paternity disputes and how to solve them through forensic dimensions. DNA evidence is used in the court to prove the paternity dispute cases, rapes and criminal cases. In paternity cases, if a particular man has allele which is strongly matching with child's allele at certain locus, then he cannot be excluded from the paternity disputes. In some cases, it also seems that close relatives will be more likely to have the child's paternal alleles. Forensic genetics have focused on the Y STR markers which are available for population genetics and forensic investigation. In this module, discussion about the paternity dispute cases, how cytogenetics is useful to determine certain cases related to forensic investigation, and the methods of DNA extraction will be discussed. The rapid development in the technology such as STR typing technology has increased power to identify real fathers, thus providing the objective evidence for solving civil and criminal cases. Cytogenetic is the study of the cell chromosome and their role in hereditary structure. The term chromosome was coined by Walder in 1888. Several discoveries were made to correct the numbers of the chromosomes. In 1955, two scientists in Sweden, Lund and Tiso, were experimenting with the culture of embryonic lung cells and established 46 correct number of chromosomes. This study was confirmed by other scientists, Ford and J. A. Hamilton, on studies on human spermatocytes. Chromosomes are 46 in number. Out of these 23 pairs, 22 pairs constitute the autosomes and are similar in males and females. 23rd pair of chromosomes are known as sex chromosome. Female chromosome are XX and males has XY chromosome in the 23rd pair of chromosomes. Later, in 1960, a group of scientists working in Denver, in Colorado in USA, adopted a system for classifying and identifying the human chromosome according to the length, position of centromere as metacentric, submetacentric and acrocentric. The chromosomes are placed according to their length. The longest chromosome are placed at starting of the order of sequences and shortest at the end. This system is referred to as Denver system. Chromosomes are divided into 7 groups according to the numbering order. Chromosomes are placed as Group A, Group B, Group C, Group D, Group E and Group F and G. In the Group A, chromosome pairs include from 1 to 3 chromosomes. Here, chromosomes are grouped according to in the position of the centromere and by the length of chromosome. This group consists three largest chromosomes. Group B, it consists of 4 to 5th chromosome. The chromosome 5 is slightly smaller than chromosome 4 according to the position of the subterminal. In the group C, it consists of 6 to 12th chromosome. Chromosome 6, 7, 8 and 11 are metacentric, while 9, 10 and 11 are subterminal. In the group T, it consists of 13, 14 and 15th chromosome. Group E consists of 16 to 18 chromosome. Group F include 19 and 20th chromosome. And finally, the group G, it includes 21st and 22nd chromosome. Now, I'll be talking about the bending patterns in human chromosomes. The technique used by staining the chromosome in metaphysics for cations are identified is called bending. A band is defined as the part of chromosome which is clearly distinguished from the adjacent segments by appearing darker or brighter with one or more bending techniques. Bending techniques are having two principal groups. First, the bands are distributed with the length of chromosome such as G band, Q band, R band and B band. Those that stains a restricted number of specific bands or structure are named as C band. These four bending patterns include Q band, G band, R band and C band. The G band is named after the Zimza dye. In G band, dark regions tends to be heterochromatic. R band, that is reverse of Zimza, the dark regions are euchromatic and bright regions are heterochromatic. Whereas C band is constitutive heterochromatin. Now, I'll be discussing the abnormal chromosomes. Human chromosomes are 46 in numbers. Chromosomal abnormalities are studied based on autosomal aberrations and sex chromosomal aberrations. The autosomal aberrations include number and structure of autosomal chromosomes. Autosome means autosomal body chromosome. Numerical autosomal aberration include aneuploidy and polyploidy. This figure is depicting different type of chromosomal aberration in men. Basically, it can be divided into two categories: aberrations which are found in autosome chromosome and aberrations which are found in the sex chromosome. The autosomal aberrations can be further segmented into numerical autosomal aberration and structural autosomal aberration. The numerical autosomal aberration consists of aneuploidy and polyploidy, whereas structural autosomal aberration consists of translocation, deletion, insertion, inversion, ring aberration and isochrome. Whereas the sex chromosomal aberration include numerical sex chromosomal autosomal aberrations. The table is depicting various kind of chromosomal abnormalities. Normally, the male's chromosomal composition is XY and the female's chromosomal composition is XX. In case of Turner syndrome, the chromosomal composition is XO. In the case of Klinefelter syndrome, the chromosomal composition is XXY. In the triple X syndrome, the female is having triple X chromosomal composition. In case of Down syndrome, that is trisomy 21, the clinical symptoms include mongoloid idiocy, and it is named after the Langdon Down, epicanthus skin eye fold, protruding tongue, short stature, mental retardation, transverse palmar crease are important symptoms of trisomy 21. Whereas in case of trisomy 18, which is also referred as Edward syndrome, the symptoms are deformed ear, defective heart, and mental retardation. Trisomy 13, which is referred to as Patau syndrome, clinical symptoms include several body malformation, small head, and small eyes, cleft palate. Y chromosome is very useful for testing the paternity. Y haplotyping application for special instances such as deficiency case in paternity testing and analysis of mixture of male and female DNA or with combination with autosomal markers. It is useful in the cases like violent crimes done by the males in sexual offences. So, at crime scene, culprits leave samples, usually informative of the Y markers in the rape cases. In the case of paternity of son, it is in question. So, in this case, comparison of the Y chromosome will exclude the alleged father. Y chromosome are found only in male sex and it passes from father to son. Y chromosome is paternally inherited and haploid. It's haploid, so it cannot recombine with other at meiosis phase. It can be done by using DNA polymorphism. Y polymorphism and Y chromosome tree consist of binary polymorphism, microsatellite and minisatellite, and haplotypic diversity. Now, I will be talking on practical applications of Y markers. Y markers are using small PCR amplicons. Y chromosome is suited for analysis of DNA samples. Characteristic of PCR is that it is large cluster of repetitive units. Y chromosome analysis for paternity testing. In the case 1, alleged father was not available. Some of the relative paternal uncle of putative son were available. These people were taken for Y chromosome marker test. Exclusion can be done on the basis if the Y chromosome is not same as putative son. In the second case, that is of rape case, semen of attacker is mixed victim cells, then sperm nuclei of attacker are separated from the component of female victim by the differential lysis of cell types. If in this case, this method doesn't give the result, then victim female profile is separated from the attacker's profile and the autosomal marker is used for analysis issued. On the other hand, in such cases of rape, Y-specific analysis allows attackers' haplotype to be determined. In case like multiple rapes, microsatellite of Y-specific allows to be determined the number of attackers. Y chromosomal markers will be very useful in analysis of other kind of mixtures such as blood saliva and blood composition. Slide number 13. Now I will be describing the structure of DNA. DNA is basically made up of long molecules that contain coded instruction for the cells. DNA contains long molecule chains. The link of the chain is in pieces and called nucleotides. These nucleotides are also referred as bases. There are basically 4 types of nucleotide called adenine, guanine, cytosine and thymine. The backbone of DNA is made up of phosphate. The diagram is showing you the typical double helical structure of DNA. The principal component of DNA include base pairs such as adenine, thymine, cytosine, guanine and the backbone, that is phosphate backbone. There are basically 3 types of techniques that are used for identification of testing. The first technique is DNA profiling, second is DNA fingerprinting and third is DNA typing. Now, you will be acquainted with the history of DNA fingerprinting. In the beginning, the identification of a person was done through the ABO blood group system. Later, new markers were discovered on the basis of the protein serum and red blood cell enzymes. Later, Sir Alex Jeffrey invented DNA fingerprinting techniques. DNA fingerprinting is a technique to determine the sequence of nucleotide in certain areas of DNA. The sequences of nucleotides are unique to individual person. Every person has unique fingerprinting. DNA fingerprints are same for every cells, tissues and organs of the individual. Fingerprints cannot be changed throughout the lifetime. Individual person can be distinguished by the entire genomic DNA sequences. The paternity determination consists of a number of steps. In the first step, the blood samples taken from the paternity case are studied by protein such as RBC protein and human leukocyte antigen typing. In the second step, DNA fragments are then cut by the enzymes restriction endonucleases. Then next, the fragments of the DNA are done by the agarose gel electrophoresis hybridized with radioactively labeled DNA probes. And finally, the size of the alleles detected by these 2 probes. Now, I'll be talking on techniques which are used for DNA fingerprinting. The DNA fingerprinting techniques consist of various steps. In the first step, DNA is extracted from the white blood cells, spermatozoa, skin and from the hair. Hair that is forcefully pulled out or natural fallen. From the root of the hair, DNA is extracted. Then DNA molecules are cut into fragments by the chemical knife with the help of restriction endonucleases enzyme. This fragment of DNA also contain the variable number of tandem repeats. On the gel electrophoresis, the fragments are separated according to the size. The diagram is illustrating the process of gel electrophoresis. In the first step of gel electrophoresis, restriction enzyme cleave the DNA into smaller segments of various sizes. These DNA segments are then loaded into wells in a porous gel. The gel floats in a buffer solution within a chamber between 2 electrodes. When an electric current is passed through the chamber, DNA fragments move toward the positively charged cathode. And finally, the smaller DNA fragments move faster and farther than larger DNA segments. After separation of DNA fragments according to the size, the particular size of the fragment which contain variable number of tandem repeats multiplied by the polymerase chain reaction technique. To split them into single strand of the DNA, they are treated with the alkaline chemicals. Then the single strand of DNA are transferred to the nylon membrane. On the nylon membrane, radioactive probes of the DNA consisting the possible variable number of tandem repeats are put. The nylon membrane are washed to extra probes. This process involves Southern blotting technique. It is named after the inventor E.M. Southern. The method of hybridization of DNA with the probes is called Southern blotting. On nylon membrane, X-ray film are exposed to probes of the DNA to mark the places where radioactive probes of DNA are bound to DNA fragments. The places are marked as dark band after X-ray film is developed. This technique is known as autoradiography. On the X-ray films, the dark band represent the DNA fingerprinting or DNA profile. Now, I will tell you different applications of DNA fingerprinting. DNA fingerprinting is very useful in identification of the human remains. It can also be used in paternity, maternity dispute, in studying the relation with the ancient humans, accused and convicted fellows. They can be set free because of DNA typing. It is useful in detection of the crime and legal pursuits, in the determination of the bone marrow transplant, in proving the relatedness of immigrations, in lineage markers, in identification of hereditary disease, and it can also be helpful in individuality of uniqueness determination. Paternity determination through allele frequency distribution of highly polymorphic DNA sequences. In this determination of paternity, blood samples are collected from the paternity cases. Sample types for blood group, human leukocyte antigens, and series of protein polymorphism for the inclusion of the paternity probability of alleged father are calculated using allele frequency. Alleged father is excluded on the basis of the size of his alleles if they are different from the inherited alleles of the child's paternity. This is done by taking each individual's phenotype alleles. Blood samples are collected, then cases of paternity samples of DNA are digested with EcoRI or TechI and loaded into agarose gel for fractionation. If there are no detectable difference between one of the alleles of the alleged father and child, then there is a probability of his being biological father of the child. Students, now I'll be describing different principles of DNA fingerprinting. Human genome consists of several small known coding and inheritable sequences of the bases. These sequences of bases are repeated several times. The sequence occur near Y chromosome, heterochromatin area, centromere and telomere. The area where sequence of DNA is repeated many times are called repeated DNA. Repeated DNA is separated as satellite from the bulk of DNA by centrifugation, density gradient, and this is called satellite DNA. The repetition of the basis of satellite DNA is called tandem. Satellite DNA shows the polymorphism segment. Short nucleotide repeats, since the DNA are very specific in each individual and they are vary in number from person to person, and this is called VNTR, that is variable number of tandem repeats. These are also known as mini satellite. This is used as genetic markers for each individual in personal identification. Next, I'll be talking on paternity testing with locus incompatibility. A case study of single nucleotide polymorphism, STR efficiency in paternity testing with locus incompatibility methods used for this test is based on the short tandem repeats, loci analysis to observe the more common incompatible STR locus. In this process, DNA samples from the paternity case were extracted, then single nucleotide protein and CSF1PO genotyping was done. Single nucleotide protein, short tandem repeat haplotype analysis, DNA sequencing, and finally done with statistical analysis. Here, observation for paternity test is that SNP-STR can be applied for paternity testing. The power of SNP-STR is much greater than the alone STR and SNP alone. SNP are also useful and they determine distinguishability between father from uncles. Now, I will tell you one of the case of paternity dispute from Hyderabad. In this case, the husband's plea is that he had no access to the wife when the child was begotten can be provided by the DNA test report, and in the face of it, court was not able to decide the fatherhood of a child. Then court decided to send them for the DNA test for scientific results. When the scientific reports proved to the contrary, and further the innocent child may not be bastardized, as the burden is on the mother to prove that the revision petitioner is the father. From the marriage also in dispute, especially of any conjugal life. While dismissing the revision petition, Justice Siva Shankara Rao ruled that the DNA test help us as a solid proof on truth of the dispute of paternity. Now, I'll be talking on immunogenetics, that is the ability to induce an antibody or cell-mediated responses. When an antigen substance which can be recognized by the surface antibody, that is B cells, it is associated with major histocompatibility molecules, the human leukocyte antigen genes, and major histocompatibility is activating and inhibitory killer immunoglobulin like receptor genes from haplotype which shows diversity. It not only reflects in only nucleotide difference of the alleles but also in terms of presence or absence in individual genes. The HLA is used to match a donor of bone marrow and blood transplant. HLA is protein found in all cells of the body. The immune system uses HLA as markers to know which cell belong to one individual's body or not. Different donor features have impact on transplant success. They include age, gender, blood type, and the number of time the female donor has been pregnant. The method used to test HLA antigens and antibodies earlier are based on the serological methods. Now, antigens are molecularly defined by the DNA analysis such as flow cytometry, solid phase immunoassay, and single antigen based assay such as Luminex. In recent year, More advanced techniques have been developed to determine the paternity. The first technique is human leukocyte antigen. High degree of discrimination of human leukocyte antigen system make more accurate probability to determine paternity. It caused the revolution in paternity determination after combining HLA with red cell antigen test. The second technique is red cell enzyme and blood serum protein by electrophoresis method. Many blood serum proteins and red cell enzymes have proven useful in determining the paternity test by electrophoresis method because protein and enzyme process follow the Mendelian inheritance pattern. So students, let us summarize what we have studied in this module. DNA extraction from biological materials such as blood group system, protein of bloods, red cell antigens, haplotypes, single nucleotide polymorphism and single tandem repeats are very useful in determining the case such as rape cases, paternity dispute, inclusion and exclusion of the alleged father. Most of the paternity test is done for the financial reason to get the legal responsibility of the legal support to the child. Such test can determine the important tools for civil court when paternity of a child is in question. Serological test for identifying a person is now an old trend. So, human leukocyte antigen typing is a good method to identify a person. Thank you.